So I'm very happy to be here, and I think my Italian is as bad as Paul's, so we'll stick to English. And thank you, Roberto, for the kind introduction. I didn't understand a word, but I hope it was good. Indeed, my team and I at the Weizmann Institute, and lately also at the DKFZ in Heidelberg in Germany, have been studying the microbiome for over 8 years. And I think it's a fascinating time for all of us studying this topic, despite the fact that this is a very young field, because conceptually we increasingly learn that with the microbiome we are introducing into the equation of the human body a new genome that includes 150 times more genes than our 20-something thousand human genes, which impacts almost every aspect of our physiology, and as you've heard in the great talks before me, almost any multifactorial disease process. And I think the billion-dollar question is to start to understand how this is all done, and this is where the field is going. Now, when I will kind of highlight where I think we stand and where I hope we go, I will start by describing to you what I think are 4 fundamental hallmarks that this very young field has reached that transformed our ability to understand microbiome research and its impact on human health and disease in going forward. The first hallmark concept, which is really fundamentally important, is that if we sequence every one of your microbiomes here in this room deeply and comprehensively enough, we would find that we all have a unique signature of both composition and function of our microbes. At the beginning, when this field just emerged, this was very frightening because it's very hard to study something that's so variable. But as we all learned and are continuing to learn, the fact that the microbiome is actually a signature or a fingerprint of each and every one of us, which is composed of a huge amount of data, enables us in many cases to use the microbiome as means of explaining different traits in healthy humans and in explaining why some humans with a genetic susceptibility or genetic risk would end up developing a common acquired disease. And I think I'll try to show you one example of our own work. The second hallmark concept is one in which we regard the microbiome, and specifically the gut microbiome, but this is true for any other microbiomes, the skin microbiome, the oral microbiome, the genitourinary microbiome. These are all could be described as a hub for signaling, which integrates signals that come from many diverse parts of the human body. So for example, the gut microbiome is influenced by the host genetics to some extent, by the host immune system, as you've heard in previous talk, but also by a great variety of environmental factors. In fact, we think that most environmental factors signal to the microbiome. The microbiome integrates all of these diverse signals, transmits them into the host, and the way we see our mission in life is to try and decode this mysterious language by which us and our microbes talk in a very intimate manner, and to see what goes wrong in this language, which leads to a risk of developing one of many multifactorial diseases. The third and very important concept, which we really just learned in the last 3 or 4 years, relates to the fact that we can imagine the microbiome also as a huge biochemical factory. The gut microbiome of each and every one of you is able to either generate or to modulate around 100,000 small molecules. Now, if I were to measure the small molecules in your sterile blood, we would find that 80% of all small molecules in a human being's peripheral blood come from the gut microbiome. And by this, you can already imagine that the microbiome, for example, in the gut, can impact processes in the gut. This is, of course, very easy to imagine. but it can also impact things that occur kilometers away from where the microbes actually live, because many of these thousands of poorly understood small molecules swim from the place where they are generated with the microbiome into the sterile host, and they can reach every cell and every organ in our body where they can impact biological processes. And we believe that the vast majority of these 100,000 molecules are potentially bioactive. So you can regard them as potential treatments or therapies. Now, just to give you a flavor of where we stand in this very young field, of these 100,000 potentially bioactive molecules which exist within your own bodies, we know the function of around 30. Okay, so there's a huge ocean of druggable components within our body which we're just starting to understand. And the fourth hallmark concept, which was elaborated before me, especially by Paul O'Toole, who really heads the global effort in trying to understand how the microbe influence aging, we are increasingly aware that one of the environmental, if you want to call them, factors that impact the gut microbiome and other microbiomes are the lifespan of a human. With the microbiome of young children, very different than that of healthy adults. And then when we get into the aging period, very interesting, very peculiar, very poorly understood changes start to appear. We know very little about the mechanisms driving these processes. But for example, in this paper, which we wrote a couple of years ago, we described that almost any of the aging-related healthy or unhealthy processes could be plausibly modulated by these changes in the function of the microbiome. And this includes frailty, which you've heard about before, inflammaging, which you've heard about in the previous talk, and also practically every aging-related disease that you can think of, for example, Alzheimer's disease, which we and many others are extensively researching in order to find a mechanistic explanation on whether differences in the microbiome between different people confer susceptibility to develop this disease on top of genetic susceptibilities that for sure play an important role. Now, the way that we see our mission in life, and actually I think that the place where this young field is going into is trying to convert from the initial phase in which we all tried to associate different microbiomes with different health or disease processes. These were associations, there were correlations, they were very important for the starting phase, but we never knew what the egg and the chicken was. We never knew what the cause and what the effect was, towards where we're going now, which is a more mechanistic, molecular basis understanding of host microbiome interactions. And if we were to break this down, we're all trying to understand how the microbiome impacts the physiology of a healthy human host. And as I said, every physiological function that you're interested in is modulated in one way or another by the microbiome. We're also interested in understanding how the human host impacts his or her microbiome. And this is an equally interesting concept and an interesting problem impacting health and disease. But specifically, we are interested in trying to decode what goes wrong in the language, in the crosstalk between us and our microbes that leads to susceptibility of developing acquired multifactorial disease. And we strongly believe, and I'll try to show you just a few vignettes of data, that by gaining such mechanistic molecular basis of understanding on host microbiome interactions, we will be able to contribute towards new principles of microbiome-mediated disease treatment. Now, I want to dedicate the next few minutes of my highlight talk into giving you my 2 cents on where we go, not only with understanding how the microbiome impacts disease, but how can we potentially manipulate the microbiome in the next decade in trying to influence disease processes in many millions of people. Now, despite all the sexiness, quote unquote, of the microbiome field and the overhype and the hand waving that is associated with this very promising field, to be fair and to be honest, when we think about microbiome-associated treatment in 2019, we're mainly talking about three non-specific and highly problematic approaches. The first is prebiotics. You've heard some examples of it. This is a non-specific, non-personalized way by which we try to use nutrition in order to impact a healthy or to drive a microbiome towards a healthy configuration. But the truth is we don't really have a consensus or true knowledge of what a healthy microbiome is. The second approach, which you've also heard about, is the replacement of a microbiome, for example, by a brute force way called fecal microbiome transplantation that is highly effective in simplified scenarios, such as hospital-acquired infections, such as Clostridium difficile, but is gradually less effective or less evidence-based, so far at least, when we move into more complex disease scenarios. And the third are probiotics, which were there before we knew there was a microbiome. But if you look at the vast amount of literature associated with probiotics, and I have, you would see that we have huge, severe problems in evidence-based medicine in proving that probiotics actually do what we hope they do. We know very little on the mechanisms by which maybe they do what they do. And in fact, to say the truth, not a single probiotic preparation has been approved by the American and European regulatory authorities as a medical indication to date. So there's many, many unknowns and problems with these currently used and very popular non-specific approaches. However, what's really promising within the next decade of the microbiome field is I think that we're starting to learn how to utilize the huge amount of person-specific data that is stored in each and every one of our microbiomes in order to progress and to develop new avenues of treatment that we would utilize this person-specific data in a more rational and more evidence-based approach towards achieving new treatments that are much more reproducible and effective. And I think that we can talk about 5 major avenues of treatment that are under intense development, and I think it would be worthwhile for you to know about them, and I'll try to give a few kind of very short examples of our own work in developing these new and exciting treatment approaches. The first avenue of treatment approach, and we've really dedicated huge amounts of effort into this approach, is based upon what I call rational nutrition, which is, taking one of the most common activities that we all do in our daily lives, and rather than trying to mix and match and guess what would be a good or bad diet, utilizing the big data and the really advanced computational technology that we can harness today, including the microbiome, in order to tell a person or to direct a person towards a personalized nutritional approach that would benefit them in a very data-driven approach. The first example that we've introduced to the world in this regard relates to a very common nutritional supplements which are used by tens of millions of people worldwide, which are non-caloric artificial sweeteners, and I'm sure all of you have tried them. I've tried them and used them many times. We put them in our food because we believe that they're inert, so they don't have, our body doesn't have the capability to break them down and to extract calories from them, but they provide us a very sweet sensation. They make our food very sweet. We like it in many cases. So this has become kind of a way for millions of people to get the sweet tooth without paying the caloric price. And to make a very long story short, what we have found was that the gut microbes, in contrast to the human body, which indeed cannot degrade these substances, the gut microbes can very easily and very massively degrade these seemingly inert molecules. And in some microbiome configurations, in some people, the resulting functional consequences actually may drive people faster towards diabetes. Now, this was very counterintuitive. This was very unexpected because many millions of us take these exact substances to avoid diabetes. But what we found was that not only is the microbiome a very potent and good explanation on why there is such a variability in clinical trials related to these substances, it could provide a mechanistic explanation on why some people actually become more prone to obesity, type 2 diabetes, and their complications when they consume them. And in fact, we could use a machine learning algorithm that would take a person's microbiome data and would enable to predict for that person whether he or she would be susceptible to these adverse effects or would not be susceptible without them ever touching or even being exposed to these artificial sweeteners. So this was the first example, to the best of my knowledge, of the microbiome enabling the prediction of a personalized response to a food component. But when we finished this project and published it, we actually realized that this was only a private example. And in fact, every single food that we ever consume in our daily lives has a personalized component or a personalized response. And this led us to a very ambitious project, which is termed the Personalized Nutrition Project, in which we took 1,000 individuals and we kindly asked them to give us a week of their life. And during this week, we've collected the biggest amount of person-specific big data from all of these individuals, including all of the features that you can see here, of course, a microbiome analysis. We connected them to a glucose monitor that measures their blood sugar levels for a whole week, every 15 minutes. So this, just to give you a flavor, this produces 2,500 measurements of blood sugar levels from each of these 1,000 individuals. We had them follow a smartphone app that enabled us to know everything that they were doing during the week, and many, many, many more data sets. So we collected this huge amount of data, and then again, we used machine learning and artificial intelligence systems in order to try and predict people's differential results in terms of their blood sugar responses to different foods computationally, and able for the first time to tailor a diet to the individual. Now, this seminal study, I think, produced three major breakthrough insights. The first is that we were able to show for the first time that sugar responses in humans are highly personalized. In other words, we can take all of you here in this room and we can give you an identical, amount of Nutella and measure your blood sugar levels. And maybe some of you would think that it's bad and some of you would think that it's good and some of you wouldn't care. But the fact of the matter is that even if you all take the same exact amount of Nutella, and your blood sugar responses would be different than one another. And this is important because if you think about it, it kind of contradicts every intuitive knowledge that we follow in our daily lives or every attempt that was made for 50 years for a one-size-fits-all diet, because if your sugar response to Nutella is opposite than mine, then Nutella cannot be part of both of our diets. And this is true for any different foods. We've now done this in almost 100,000 people and have quantified the data from millions of meals. And basically, this individualization is true for every given food. Now, more excitingly, we found that we are able to take a big data set from a given individual and use machine learning in order to predict that person's response to any given food. So for the first time, we can take a person, measure that person, and tell that person what foods are good or bad for him in terms of their sugar responses. Again, this has now been reproduced worldwide by other studies. And the last exciting knowledge that we learned from this study is that now that we can predict a person's response, if we take a new cohort of human individuals and give them or let them eat their person-specific bad or good foods, we can impact in a very reproducible way their glycemic responses. Now, this is not easy because if you know the literature or the medical field, the obesity and diabetes epidemic is perhaps the biggest epidemic in our people's lives, and we've so far miserably failed to form any nutritional intervention that would be good to the billions of people that suffer from this epidemic. Now, you can see here an example from one pre-diabetic individual. That individual already have spikes of blood sugar levels after meals that are almost in diabetic levels. And now you can give this individual a personalized diet, and you measure for a whole week their glucose, their sugar responses in green after a week of a person-specific diet that could in some cases include ice cream or Nutella or whatever. You can never guess what is good or bad for you until we measure you, and we can completely normalize even in a short-term intervention, your glycemic responses. Now, this was very nice for us and kind of flips the entire paradigm on its head, but of course, it also begs the question on whether this intervention could be worthwhile in the long term. And this led to a very ambitious study, which we just completed. We haven't published it yet, in which we had a double-blind kind of approach in which half the people, 200 people, followed this personalized dietary approach. The other control arm followed the best diet currently available, the American Diabetes Association diet for a whole year. So this is a long-term study. And we wanted to look at many different metabolic parameters to see whether our dietary approach is actually superior, perhaps, to the best diet offered to these pre-diabetic individuals. These are the results, which again are unpublished, and you can see that whatever objective measure that we measured, the hemoglobin A1C, we measured them by a continuous glucose monitor for 6 months, never done before. You can see that the glycemic levels in the individualized dietary arm are dramatically better than the current gold standard. You can compare them to even to medical treatment in previous studies, such as metformin, and to the best of our knowledge, this personalized approach is superior to all of them, and hopefully we'll publish this data soon. We also, of course, measured many other metabolic parameters, and you can see, for example, the triglycerides are significantly lowered with just this dietary approach. With any other component added to it, and HDL is increased. So this, to us, is quite reassuring. We are also engaged in many other follow-up studies. One of the biggest unmet needs is childhood obesity. In the US, close to 20% of all children are officially obese, and they start to suffer from fatty liver and diabetes in very young ages. And as part of an effort to utilize this personalized approach in the children population, we are engaged with a group from Naples in trying to see whether we can adapt the personalized nutrition approach to the pediatric population, and whether we can learn to modify diet in a way that would not, of course, harm their growth and so on and so forth, while avoiding this childhood obesity epidemic. So this was the first major avenue of microbiome research, which we and now many others are following. The second is very intuitive to all of you. It is composed of microbiome or microbial transplantation, in adding a bacteria to the microbiome and trying to improve our health. And of course, the first thing that comes to mind is probiotics, which we've used for decades. We didn't even know the word microbiome when we started using them. As I said at the beginning of my talk, evidence-based in many of the indications is not sufficient, to put it mildly. We had our modest contribution to the field by performing the most invasive microbiome study ever, in which we, in different phases, took many individuals and scoped them twice before and during probiotic consumption, in order to study for the first time directly in the gastrointestinal tract what happens when we take exogenous probiotics and put them into our system, which already contains trillions of very well-fit microbes. And to make a long story short, we were surprised to find that around 50% of the people we examined featured a very personalized resistance to these exogenous probiotics. We've tested 11 of the most common probiotics that you can buy in your local supermarket. And in these people, you take the probiotics from one end and they quickly reach to the other end without ever colonizing the host or having any measurable response induced in the host. In the other half, the microbiome, for some reason, enabled the colonization of these microbes, and there we could see an effect. So just like with food, we found that colonization of probiotics is highly personalized. And again, by using machine learning, we are able to take a baseline microbiome of a person and tell him or her, based on this machine learning algorithm, whether they would be resistant or permissive to the probiotics they will be taking, even before they ever saw the probiotics. In another component of the study, we also highlighted unknown adverse effect that may be associated with common prebiotic consumption. For example, when you take them with antibiotics, they may